perhaps. Although you can never say never, but it's very, very difficult, to say the least, to do full-related screens in the mouse for development. The development is internal, it's difficult to obtain mutants, and the generation time is not appropriate. So flies and worms were used initially to isolate mutants, and remarkably, the genes and the concepts that were delineated and described in the fly and worm systems, among others, not exclusively those systems, but they're very important, proved to be conserved and therefore applicable to mammalian vertebrate, first of all, and mammalian developmental genetics. And that allowed scientists to actually study development in the mouse through reverse genetics. So what's reverse genetics, you've seen that term before, is you can take a gene in the fly and find the ortholog in the mouse genome. And amazingly enough, well, now it's not too surprising, at the time it was, you can find such genes, such orthologs in the mouse genome, and you can make mutations in the mouse system, do conditional deletion with a preload system, or even induce point mutation in the mouse system. How did you know what gene to focus on in the first place? Because these genes were already identified as genes important in development in the fly or in the worm. So there's just a very important interplay between organisms here, and it really shows how important it is to use the right system to study the right pathway. So that's why flies and worms, and now the fish, I would say, are really essential and invaluable system which study development. So in general, what you deal with here is, like I said before, cell differentiation, pattern formation. And so this, just to give you an example, to show you what's causing these mutations and these phenotypes. But see, when you have two pairs of wings here in the fly, this is actually a double mutant fly, bithorax, post-bithorax. These are the two mutations that generate that phenotype. This is not a gene that's doing something very specific. These two mutations don't really control, the amount of keratin that's produced by a cell. Okay, these are very, very high in the hierarchy of gene expression, and these genes, when mutated, okay, they don't in fact impact on on how the cell does something. It impacts on what the cell does and how the cell differentiates. So these cells, these animals that mutated for these genes, produce a perfectly fine pair of wings. You're not mutating a gene that's important to produce this shape or the molecules part of the wings. It's a program mutation. It's called a homeotic mutation. We'll get back to that. And same idea, here's another impressive phenotype. It's a mutation called antinapedia, very famous example, where the mutant fly, this is a single dominant mutation, grows a pair of legs instead of the antenna. So same idea, it's a program mutation. That leg is perfectly fine. All the hairs and the muscle and everything is done just the right way. But that cell is mistaken on its identity. It's differentiating, and this group of cells actually differentiate another structure. It's called an appendage in this case. So in general way, genes in early development control are the recognition of the position of the cell in the embryo. So the real problem in developmental biology is you start with one cell. At the end of the day, that's what fertilization is. It's a single diploid cell which will divide into 2, into 4, etc. So how come from that single cell you generate these vastly differentiated cell territories? That's really the question in development. And sometime during early embryogenesis, there is formation and differentiation of specific axes in the embryo. So you have the antero-posterior axis, you have the dorsal-ventral axis, and the left-right axis. So this cell right here has a certain coordinate, okay, versus X, Y, and Z. And the one close to the tail has a different coordinate. And this is the thing, as the embryo develops, there is an establishment of symmetries, left, right, and of axes, dorsal, ventral, and antero-posterior, which will determine the differentiation of each cell relative to its position in the embryo. How are these axes produced in the first place? Well, the answer is the production, very broad answer, we'll look in the fly how this works, is the production of a symmetry at the one cell stage in the oocyte. And what you should know, a very important concept, is that a given master regulator gene, such as the gene that's important for production of the appendage, such as the wing or the leg, actually that same gene can lead to different expression programs depending on the position of the cell in the embryo. So this is not a situation where a gene is going to say, okay, you're going to make a leg. It's more like a gene is going to say, you're going to make an appendage to a group of cells, and depending on where that cell is in the embryo, that appendage can be an antenna, can be a wing, can be a leg, things like that. But that gene product is the same. So how can a master regulator have different outcomes depending on the position of the cell? And this is what I want you to remember, is that cell that's responding to these activities has already differentiated and therefore has different responses to a same transcription factor. And so a same transcription factor will activate transcription of different batteries of genes in the cells, in cells, depending on where they are in the body and in the embryo. In general, this is kind of different categories of issues that are important in developmental genetics, and how these asymmetries and differentiation of cell territories are controlled. There are essentially different set of activities. So it's not like one is happening in one system and not the other, or at one time in development and not the other. All of these mechanisms occur pretty much everywhere at all times. So we're going to discuss specific examples regarding these mechanisms. One is, as we just talked about, the reason why cells differentiate differently depending on disposition is called positional information. And I'll give you the example of how that works in the fly. Positional information requires the response to a gradient of a specific molecule called a morphogen. So I'll tell you what a morphogen is, and I can tell you now, morphogen, in fact, we'll reiterate, is a signaling molecule that induces specific batteries of genes depending on its concentration. So a gradient is produced, And the way this works is that, in fact, the response to the morphogen is extremely steep. That is, a very small difference in concentration of the morphogen leads to a quite impressive difference in the gene expression program that is elicited. And so this is how cells that are close in the embryo can have vastly different outcomes. For example, in the anterior pole, cells respond to the morphogen and they develop in the fate called blue, for example, but cells that are very close to the posterior side of the blue region actually may have a completely different developmental program in response to a relatively small concentration of morphogen. So that's one way that differentiation occurs. It's a very steep response to the gradient of the morphogen. So for that reason, this is called the flag model, or the French flag model, because this is a system or an example of a very different territories, if you will, of areas that are close to each other. Then there's another group of interactions, if you will, it's called induction, when a single cell produces molecules to which neighboring cells respond, and they differentiate in a specific structure. The third set of interaction is called inhibition. It's kind of the reverse side of the story. Is say a single cell can produce a molecule that diffuses nearby and will inhibit neighboring cells from differentiating with a particular fate. So these are the three important types of interactions occurring in development: positional information, induction, and inhibition. And so for that reason, you see that development relies heavily on diffusible molecules, and these cells responding to morphogens, responding to inducing or inhibiting factors. The neighboring cells here require a receptor to that molecule, and they need to respond to it, and that is done through specific signal transduction pathways to respond to these molecules and to induce the proper and timely transcription programs. And now you understand what I was telling you in the first slide. That's why when you study development, you're really studying a lot ligand receptor interactions and signal transduction programs and pathways responding to an external factor such as an inhibitor or an inducer. So now after these generalities, we can dive into our first model system. We're going to talk about the fly now, and then I'll talk about this today. And next lecture on first day, we'll talk a little bit about the worm system. So I want to tell you a couple of things about C. elegans as a genetic system as well. So the development of the fly is shown here on your left. That development, depending on the temperature, takes 9.5 to 11 days. That's about the generation time. So you see it's a fast generation time. And the number of progeny is in the hundreds, as you know. And so the female actually, I just noticed that. It looks like the male is laying the eggs, but of course that's not exactly. It's pretty funny. Okay, anyway, the female. In fact, the female is a little bit bigger than the male, so this is correct. But I think they should have put an arrow there anyway. The female lays fertilized eggs, which are of course single cells. And I'll tell you what happens during the first 3 hours after fertilization, because that's the time when pretty much the body plan is laid out. So I see it's these events of establishing the axis that I was telling you about are extremely fast after fertilization. So after that, you have 3 larval stages that are motile, and those larvae look for food and crawl around in the food. And these are called first instar, second instar, third instar. And the third instar then finds a place in which to develop a pupa. So that's the pupal stage. This is, of course, immobile. And that at that point, the pupa secretes kind of a hard shell, and that's on day 8, and that lasts for a day or 2. And this is where metamorphosis is occurring, and then the adult fly flies away from the pupa. I'm sure you've seen, other examples of that. But here's something the book is forgetting to tell you, is that metamorphosis, I'm going to tell you now, but it's not in the book. Metamorphosis involves a huge amount of cell death by apoptosis. And so the tissues that are part of the adult, that make the adult flies, actually originate from small groups of cells in the larva. So you can actually dissect this larva here. You take the third stage because it's bigger, and you can see in this larva small groups of cells, and they're called imaginal discs. And these small group of cells, there's about tens of them, 10 or 15, I don't know the exact number, but you'll find the imaginal disc for the wing, you'll find the imaginal disc for the mouth structures, for the legs. And these come from a small group of cells that are identifiable in the larva, and the rest of the cells die off. They're not part of the adult. So metamorphosis involves the proliferation and differentiation of the cells that are part of the imaginal disc. But at that point, everything is said and done. The cells on the eye imaginal disc will form an eye. There's no way back. Okay, so what we're going to try to think about is early events. What are the earliest events that define the symmetry or answer to this fascinating question, which is how do we get such complex structures from a single cell, which is the oocyte, this single cell here. So let me tell you about the first 3 hours now. So this is essentially the fertilized egg with the diploid nucleus. Visually, you can actually tell which is the anterior-posterior pole. And this is a single cell in which the nucleus is going to divide something like 900 times, okay? And to form hundreds of cells. It's going to divide, in fact, exactly 13 times, say, from the single nucleus to the structure that we're going to call the cellular blastoderm, just shortly after 3 hours, like 3.5 hours. So this is actually, the whole thing is 13 nuclear divisions. Now, in the first 9 nuclear divisions, okay, the nuclei divide, but the cell does not. So all these nuclei are dividing and are sharing, say, the host cell, if you will, and there is no cell division at this point. So we end up with a single cell that contains something like 9,000 nuclei, somewhere around here, and that cell that contains these thousands of nuclei is called a syncytium. And that's very important to remember, that all these nuclei share the cytoplasm, and that structure is called syncytium, right here.
After 9 nuclear division, that is just before the 10th nuclear division, the nuclei migrate towards the periphery, and you get a structure, as you can see here, with the nuclei forming a single layer around the periphery or the cortex of the embryo. At this point, that's still a single cell. However, you have something like 10 or 20 cells that have migrated to the posterior pole and form what's called the well-named the pole cells, and they are individual cells. And these pole cells will, in the adult, form the germline, that is the oocytes for female and the sperm for the males. So the germline is actually separating itself earlier than other cell types during embryogenesis. I spoke about these cells before, if you remember, when I talk about P elements and production of transgenes. And these are the cells in which you inject transposase on the P element. And these are the cells that actually can undergo translation. What's a syncytium? It's a single cell that hosts several nuclei. That's produced by nuclear division in absence of cellular division. Any other questions? Once the cells have migrated to the cortex and the pole cells have separated themselves to the posterior pole, there is a process called circularization, which is fascinating in itself, in which the membrane invaginates between the nuclei, so you're getting separation of each individual nuclei, and each individual nuclei gets essentially encased by membranes. And there's a single layer of nuclei here, and that means that the cellularization process, which leads to the formation of a single layer of cells around the periphery of the embryo. Eventually, this gives you, there's actually a few more cell divisions after this happens, and so you have, I think, 6 layers of cells here, and this leads to the production of the cellular blastoderm. Once again, just after about 3 hours plus a few minutes of development. And what you can see on the larva, which I'm going to talk about later as well. So here we look. This is a larva that's like a day 4, probably looks like, but this can be seen in the second larva as well. So much later, this develops as a larva, and you can see that the larva is segmented, and I'll talk about this later on. So we have specifically 3 thoracic segments, T1, T2, T3. Again, these contain the imaginal discs that will form the thorax. And 8 abdominal segments, A1 through A8, which can be visually determined by just regular microscopy, light microscopy inspection. There are also, to the aficionado, head segments, and they're actually, technically speaking, there are 3 head segments, but you don't see them easily. Okay, the reason to differentiate them is through more like molecular analysis. So the body plan of the fly is 3-3-8, 3 head segments, 3 thoracic segments, and 8 abdominal segments. There is gastrulation shortly after the formation of the cellular blastoderm. So a few hours after the production of cellular blastoderm, you get this. And you actually still, you see the segmented pattern here after gastrulation. You see the head segments have increased in size, and the thoracic segments T1, T2, T3, and the abdominal segments, which after metamorphosis will give rise to the structures here on the fly. The head containing antenna, the eye, the mouthpiece, and the labial structures. There are 3 pairs of legs on each of the 3 thoracic segments, T1, T2, T3. The wings and the alveolus on T2 and T3. We'll talk about this later on. And the abdominal segments, A1 through A8, do not have appendages. So now that we've described, say, the general structure of the body plan and the embryogenesis, how does this happen? Well, I'm going to insist on the morphogen that controls the anterior-posterior axis in the embryo, and this molecule is called bicoid. So the nuclei divide in the syncytial blastoderm, and like I said, they share the cytoplasm, but qualitatively, at this point already, a nucleus that's in the anterior pole is, in fact, experiencing a different concentration of the morphogen than cells that are in the posterior pole. So you see, it's very quick. And that morphogen is called bicoid. So before I go on, let's go through a first broad view of development. Because what we're going to see here is that development occurs, as I just told you, in specific stages and is orchestrated by a hierarchy of genes that can be classified in different categories. So you have category 1, which are act early, category 2 later, and etc., etc. So in all, we define 5 categories of genes that act in a hierarchy. First, the category 1 genes have to act, and then sequentially the different categories of genes are activated.
Bicoid is a product of what exactly?
Bicoid is a morphogen, and it's a product, it's a gene product of a gene called bicoid, and I will tell you more in a minute or two.
Sorry, you might get to this, but is bicoid in the cell, or is it outside?
Bicoid is in the oocyte, and I'll tell you more just a minute. I know, it's fascinating. I was just trying to get you interested in it, but I'll tell you more about it. But first, I want to tell you, though, about these different broad categories of genes. The first category is called coordinate genes. These are the genes that are important for this sensing of the X and Y and Z coordinate. And bicoid is one example of these genes, of the coordinate genes. And so, mutations in these class of genes actually leads to a complete confusion of what's anterior, what's posterior. So you get mutants that actually have no heads, but 2 different tails, for example. More on this in a minute. Following the activity of the coordinate genes, we have a class of genes called GAP genes. And you see they are expressed in broad regions of the embryo, such as Knirps or hunchback. And, oh, I need to tell you, the fly genes, in case you haven't noticed, they have really funny names. There's hedgehog, gooseberry, and others, here's cappuccino. There's, no, I'm serious. There's a gene called Pentagon. And, you know, that probably comes from the Morgan era where they decided, you know, this gene is going to be called white based on the phenotype.
or yellow based on the phenotype. So phylogenesis accumulated mutations, and they decided, like for example, I'm going to tell you more about this gene as well. It's very important. This is a mutant that is very different from wild type, and it has all these hairy segments that are lumped together. And so Eric Visha said, that looks like a hedgehog. So we're going to call this mutant hedgehog. Nowadays, people tend to call their gene, XRC3, dash 2B. It's not as fun. But more on the hedgehog, because that's actually the one gene. Well, I'll tell you now, the homolog of a hedgehog in the mouse is one of the very rare genes in mammals that's actually has a funny name. It's called sonic hedgehog. And you know what sonic hedgehog is? I'm the only one who didn't know this. You know what sonic hedgehog is? It's a video game, right? Did you know this? We are like too old for this. So anyway, let's back to this. GAP genes, they are activated by coordinate genes, but here, and it's really an amazing thing that's happening, you get a more complex pattern of expression. It's not like all at the head or at the tail, at the posterior end. This you get like broad territories that define, say, the first, T plus A1 through A6 segments, or things that are expressed in the middle and at the end, hunchback and Knirps and other like giant and things like that. They have more complex patterns of expression. So how can you get that asymmetry as a result of the action of the coordinate gene? We're going to touch on that question. And then, following the activity of the GAP gene, you have a set of genes called the pair rule genes. And these are genes that are expressed in every other segments. So I have a gene called even skipped, and as its name indicates, it's expressed in the odd segments, and then the one that's called odd skipped is expressed in the even segments. So these are the mutations that, or the mutant phenotype that that result from mutations in these genes. And once again, I'm reminding you what I told you before. Say, if even skipped is expressed, for example, here, right? It doesn't mean it's activating the same genes than over there. Because the genes in T3, the cells in A2, have already differentiated at that point. They respond differently to even skipped or to odd skipped, etc., than the cells in A8. That's an important thing to remember. Following the activity of the pair rule genes, there's a class of genes called the segment polarity genes. And these are expressed, in fact, in half segments. So here you see you have whole segments that express these genes and gene products in segment polarity, then, and hedgehog is one of them, and gooseberry and others. They are expressed in half segments, and they determine the activity of a stripe of a segment. Okay, so we are subdividing essentially in terms of gene expression areas as we go along. And the cells in smaller and smaller areas will express specific master regulators of transcription and will, depending on where they are in the embryo, respond differently to these gene products. There's a fifth category that's not here, but we're going to talk about the homeotic genes. These are mutants I showed you before with two pairs of wings or an antenna, leg instead of the antenna, and that stands from the Greek for similar to. So homeotic genes
homeotic mutations lead to the establishment of a completely different program for what's called a parasegment, which is half a segment, and they are actually activated by segment polarity genes. So there is this cascade, this hierarchy between these different categories. So you really must understand and remember these genes, the gap, the coordinate genes, the gap genes, parallel genes, segment polarity, and homeotic genes. That's the order, the sequence in which they act. How do you get mutations that affect development? Here's the conundrum. You want to isolate mutants in the fly that govern development. Easy. I mutagenize with, X-rays or things like that. And I cross the flies and I look for embryos that are dead. And then what do you do? So you see the genetics here? You have to be able to isolate these mutations, and you have to be able to do process with these mutations, and you have to be able to maintain this mutation in a line of flies so that you can clone the gene that correspond to that mutation. But at the same time, you want mutations that block development. So this problem was solved by two brilliant fly geneticists called Eric Wieschaus, who's at Princeton University. I'm pointing towards Princeton, and the other one, I guess, east from here, is Christian Nussmann-Volhard, who's at Heidelberg in Germany. And what they reasoned is that really the category of genes that we want are genes that in fact affect maternally supplied products that are necessary for the early development of the fly. So they did not look for what you call zygotic mutations, which are mutations in fact that control the development of the animal that carries those genes. They actually screen specifically for so-called maternal effect mutations. So those females were fine, they could develop, but they were homozygous for maternal effect genes, meaning gene products that were supplied to the embryo. And that's a strange idea, because it's not something that would come up just like that. It turns out that Wieschaus and Nussmann-Volhard knew something very well, is that in the first few hours of development that I described, this syncytial phase, it turns out there's very little transcription going on. The mRNAs in that early embryo are actually supplied by the female. So it is possible to obtain a homozygous mutant female which has parents that are heterozygous for a mutation, so everybody develops normally to term and makes an adult. But that female it doesn't have wild-type mRNAs to supply to her eggs, and her embryos then are affected. And it's in her progeny, in that female's progeny, that you detect an arrest in development at a specific point in development. So all of the progeny of these mutant females will show a specific phenotype, which is that the progeny, the embryos that are laid, all arrest at a specific stage in development. But then you have a line to work with. You don't care about the embryos, if not for the phenotype. You care for the phenotype. But then you can propagate that mutation by taking a heterozygous sibling of that female, and then you know how to use balancers. So that heterozygous female can actually propagate the mutation, you can work with it and clone it, and so on. Now, this leads to a little bit of interesting genetics. So look at this table. So this is just the regular zygotic genetics as we discussed it plenty of times, and that actually doesn't work in our case. So see if you have a heterozygous parent, right, you get homozygous mutants and they're dead. Okay, that's like just regular zygotic mutations as we discussed them before. This is how maternal effect genes are isolated. The phenotype of the offspring does not depend at all on the genotype of the offspring, but on the genotype of the female. So what you're trying to obtain here is a homozygous mutant female, and whatever you cross it with, homozygous wild type, heterozygous wild type, or homozygous mutant, doesn't matter, because that female that I'm circling here does not have productive maternal mRNA to supply the oocytes. So all the progeny is mutants, no matter what the genotype of these animals, of these embryos, actually is. Then you'd say, how do you get such a female? Like I said before, this is actually obtained by crossing two heterozygous animals, male and a female, and then the homozygous mutant develops to term because her mother, the female in the previous generation, is heterozygous, so is able to lay eggs that will go to term and that will produce an animal. But any kind of heterozygous situation for the female leads to a progeny that is all normal. So that's how to obtain mutations in genes governing the early aspects of development, even if they're embryonic lethal. You're able to isolate a line that possesses such mutations. So that class of mutations does not affect the development of the animal, the female in this case itself, but of its progeny. But in a sense, these mutations confer sterility. How does that work? So I posted a picture that I took from another book, which I think is useful to understand this concept of maternally supplied mRNA. So this is just for your edification, just for you to sort of picture how this works, because that I thought was kind of missing. So this is how, I'm sorry, it's kind of really dark, but this is how oogenesis works in flies. It turns out that the oocyte, that is actually the product of meiosis, is a descendant of a cell that has undergone 4 divisions. So you have 16 cells, and one of these cells, in fact 2 to be exact, but one of them will not form an oocyte. But the one that forms meiosis and forms the oocyte actually keeps cytoplasmic bridges and canals, physical contacts with all the other cells. So all these other cells are diploid cells that have not undergone meiosis, and they're called the nurse cells. And in this pinkish color here, that's the oocyte that's developing and ready for fertilization. So these nurse cells are the ones that are transcribing the mRNAs and are supplying these mRNAs important for development of the oocyte after fertilization, so of the egg after fertilization. And you can actually see microscopically, it's a really beautiful picture. I'm sorry it doesn't come out. I hope you have on your file, it looks good, that you have these, you can actually see these canals. This is actin staining. You know, actin is important for that. You can actually see several of them between the oocyte and several nurse cells here. So the basis for maternal effect here is that maternal mRNAs are transferred to the developing oocyte through physical connections called canals, and these RNAs are important for the development of the embryo early on. And zygotic transcription actually, so that is the gene of the embryo itself, become important somewhere around 3 to 4 hours after fertilization. So transcription starts kind of later on, after the blastoderm stage, and that's when the genes of the embryo itself take over and become important. So are there questions about this? Because I know that's not easy to think about. There's actually in your homework, there's a question on that. So remember one thing when you see that question. When you see that question, remember that really the essential thing to remember is that everything depends on the genotype of the female laying the eggs, not of the embryos themselves. And such a female must result from a cross of 2 heterozygous parents. That's the only way.
So these nerve cells, do they produce any morphogens?
Do these nerve cells produce morphogens? To my knowledge, not for themselves, but your favorite product, Bicoid, actually the mRNA is produced by these nerve cells and transferred into the egg. So, but Bicoid is not translated in the nerve cells. Only the mRNA is made and transported into the oocyte. Translation of these mRNAs occurs only in the oocyte. Does this apply to human development? Actually, this does not apply to human development. There are other molecules that are important for polarization, but this does not apply, and the actual mechanism of polarity is different in mammals, but the genes that govern development are similar. So the exact modality of early development is actually different. But this notion of master regulators in gene expression program is highly conservative. Now, Bicoid. So I actually said something, in fact, in response to your question. Bicoid mRNA is made in the nerve cells and transported into the oocyte. Once the mRNA for Bicoid is transported into the oocyte, something remarkable happens. And that was the first example of such a phenomenon. It was for Bicoid. Its mRNA is actively transported towards the anterior pole. And the mRNA for Bicoid accumulates at the anterior pole. Now we have a handful of examples where mRNAs are actively transported this way. But for Bicoid, that completely broke a gigantic rule of gene expression, which is, of course, that mRNA isn't produced and a single cell is going to be homogeneous with all these components. Not so. What happens in the oocyte is there are motor proteins that will bind Bicoid mRNA and transport it. As you can see, this is the mRNA to the anterior pole. And in all of this discussion, I really want you to pay attention to what is mRNA and what's protein, not the same thing, okay? So be careful. We're talking about the mRNA at this point. So the maternal mRNA is actively transported from the nerve cells into the oocyte, and then the maternal mRNA is actively transported to the anterior pole of the oocyte. And there, Bicoid then is translated, and of course it's going to be translated initially at the anterior pole, but then the protein can diffuse, right? Can diffuse a little bit from the side of synthesis, and that's what produces
the gradient from the anterior pole to the posterior pole of bicoid protein. So the concentration of bicoid is high at the anterior pole and is very low at the posterior pole. And this is one of the mechanisms for the establishment of polarity in the fly head. In other words, simply, what you think is a single cell actually is not homogeneous. That's what it is. So bicoid is a maternal effect gene. It encodes a morphogene. We'll talk about this in a minute. That's the definition of the morphogene. I already told you it's a molecule produced and that forms a gradient and that directs developmental phase depending on its concentration. And that accounts for the French flag model, where you have a very small, the response to very small concentrations can be great in terms of gene expression, depending on the position in the embryo. And then you have a gradient along the EAP axis, and I told you this, and now it turns out we know bicoid is essential for differentiation and formation of head structure. So it's an anterior differentiation activity. And so you can overexpress bicoid in the fly, and the head segments, those 3 segments that I told you about, actually take a larger volume in the animal. And you can limit the amount of bicoid that's produced, and T and A, thoracic and abdominal segments, will actually take a larger volume than normal, and the head will be very small. You can lose bicoid activity completely, and what you'll see is a complete disappearance of head segments, and essentially there's no anterior pole. Okay, you have 2 posterior poles. That's kind of the total loss of function. You can inject bicoid, which is kind of the same as overexpression, but at a specific location. That's an interesting experiment, because you can actually generate, in doing this, you're generating a gradient abnormally, say, over here, like at this point of injection, and you'll have a head segment that forms right there at the site of injection, and then you have the first thoracic segment, and etc. So really, you see, you can fool the system by simply enforcing the production of a different gradient, and that shows that bicoid is an activity that is important to form the head structure, and that allows the recognition of the anterior pole. So bicoid has many activities. Importantly, it's a transcription factor. Okay, and it's a transcription factor for a gene that is a little bit more complicated than what just tell you. So I'm going to tell you now. This differentiation between like the coordinate genes and the gap genes and the parallel genes and all this, some genes are a little bit in the middle. And it turns out there's a gene called hunchback. And hunchback qualifies as a maternal effect gene because its mRNA is transported in the egg, just like bicoid. But unlike bicoid, hunchback is not selectively transported. So it doesn't form a gradient. So if you look at the mRNA, I insist it's the mRNA, and you have on the y-axis quantity, and on the x-axis position in the embryo, that's the anterior pole on your left and the posterior pole on your right. I told you that bicoid mRNA accumulates at the anterior pole. Hunchback is produced by the nerve cells, but it's not transported, so it is homogeneous, okay, in the cell. But it turns out bicoid has an effect on hunchback. And bicoid is able to activate transcription of the hunchback promoter. So hunchback, but that's a little bit later in development, is one of the first genes to be transcribed zygotically. This is zygotic transcription produced in the embryo. And that's under the effect of bicoid. And Hunchback has a number of different binding sites in its enhancer for bicoid. Okay, has more than 3, but just for the sake of the argument, we should say it has 3. So that means that Hunchback is going to be actively transcribed and produced where bicoid is high in expression. So that is the anterior pole. And indeed, that's what you see. Hunchback has a very high expression pattern towards the anterior pole. That's because, despite the fact that the mRNA is early on not localized or not particularly transported, its expression under the action of bicoid is highly polarized. See? It's high where bicoid is high. But as you know, what scientists do is they say, it's not, what we can do is to mimic the position effect here. We can reduce the number of binding sites for bicoid. And if you reduce it from 3 to 1, you see the amount is lower and lower. And Hunchback is not produced as far to the posterior pole. And this is actually one element of the French frag model, because the Hunchback promoter actually responds in a great way to small difference in concentration of bicoid. And so, in fact, there's a threshold issue for bicoid to activate Hunchback transcription. There's a point at which transcription is high, and then a little bit farther towards the posterior pole in the embryo, the transcription is very low. You can barely see it. Okay. And the protein production actually falls off quite rapidly in the embryo, as you can see here in the wall type. So this you can actually see if you look at the protein profile in this case, and you see from an mRNA that's not localized, you actually get production of the protein in a polarized manner under the action of bicoid. There's another gene product that affects the expression of Hunchback, and that's another maternally supplied mRNA called nanos. And nanos is also actively transported, but this time to the posterior pole. You see that at the top graph. Very easily see nanos is high at the posterior pole. And nanos is a translational repressor of bicoid. And in fact, even if you have bicoid RNA in the posterior pole, nanos in fact represses the translation of this maternally supplied RNA. And therefore, the protein, Hunchback protein, is low at positions where nanos is high enough to repress the translation. And this also impacts on the profile of Hunchback production in the embryo. So Hunchback mRNA is maternally supplied, doesn't form a gradient. It's positively regulated by bicoid for zygotic transcription and translationally repressed by nanos, itself also maternally supplied. And this is how you get this kind of expression pattern. So now we talked about maternal effect genes. We just moved a little bit into gap genes, because in fact, the first category with bicoid and nanos, which are maternally supplied, you see how they impact on the expression of, say, a downstream genes such as Hunchback, and those are the gap genes. And these are the genes that will localize to larger segments of the embryo. And so that's something that's remarkable, because you see that when it's easy to picture that bicoid or nanos are expressed at the pole, because all you have to do, well, easy to say, but all you have to do to explain this is to move the mRNA. But how do you get an expression pattern straight in the middle here? It's difficult to explain. And this is an example of a gap gene called Kruppel. And here's the bottom line. Kruppel is turned off, in fact, translationally by Hunchback, and Kruppel is turned on transcriptionally by Bicoid. So I told you where Hunchback is and why it's highly expressed in the pole. You have no Kruppel there because of Hunchback. But there's a zone in the embryo where Hunchback is still fairly low, so there's possible expression, and Bicoid is high enough, let's say somewhere around here, where Hunchback is actually reduced, has reduced amounts. So, and there's enough Bicoid to induce transcription of Kruppel. And Hunchback is still present, but not to an extent that it completely needs translation. So you see that you're, with these relationships, these regulatory relationships, you are able to explain patterns, more complex patterns for gap genes expressed in kind of broad regions in the embryo, such as the middle zone or the abdominal zone, etc. So this is another example. These are more complex regulatory interactions, giant, and these are gap genes that regulate their expressions. And you see, giant also regulates Kruppel. Okay, so there are many more interactions to describe this. So gap genes are transcriptionally activated in discrete domains of the embryo, and you start seeing now more complex pattern of expression rather than just gradients from the interior and posterior pole. So now we can talk about pair rule genes. Before I go there, I want to go flash this picture from a later image. Just to explain one thing is, I told you that the larva could be seen as a number of segments, T1 through T3 and A1 through A8 in the head segments. It turns out that at this point, the real zone of expression that's relevant for development is not really the segments, because these are more like morphological entities that we see, but the relevant zones that are controlled by gap genes and later on by pair rule genes is a zone called the parasegment. And the parasegment is simply something that kind of falls in between two segments. So for example, you have the segments listed down below. For example, parasegment 4 has the posterior side of T2 and the anterior half of T3. So it falls like this in between two segments. And in fact, what you'll see later on is that parasegments, in fact, by segment polarity genes, which we're not going to discuss today, are further delimited into two halves. Okay. So the gap genes control the expression of pair rule genes as stripes. So in other words, gap genes are a regulator of transcription and expression of the next category in the hierarchy called pair rule genes. And for example, even skip forms 7 stripes in the embryo within 14 stripes of gene expression called parasegment. We have 14 parasegments. And for example, even skips forms 7 stripes of expression. And that's under the control of gap genes, and I'll tell you a little bit how this works. And each parasegment overlaps with 2 segments. So let's talk about pair rule genes. So now what we have is back to maternal, coordinate genes and gap genes, we have a very complex pattern of expression of tens of genes at this point. Giant, crouple, bicoid, etc. And now we have parallel genes that have extremely complex sets of regulatory regions and their promoters. And these regulatory regions respond to different sets of gap genes, including bicoid, which has lingered along for the ride. Now, so here's the pattern of expression of even skipped. And you see I have 1, 2, 3, 4, 5, 6, 7 stripes. And these delimit parasegments. And the gene is under the control of a number of different sets of regulatory regions, which are combinations, very complex combinations of enhancers and silencers. And the fascinating thing is that in each stripe, the expression is controlled by different regulatory regions. And that has to do with chromatin modifications. So for example, in stripe 2, this is the element that controls the expression of even skip in stripe 2. If you take this little piece and put it under the upstream of LAC-Z, that will turn blue. Only stripe 2 will turn blue, not the other stripes. And if you mutate that, you will not get expression in stripe 2, but you'll get expression in stripe 1, etc., etc. For example, if you take stripe 5, that's the element over here. Stripe 5 requires this piece, but not the others. So this thingy, which contains, a bunch of enhancers and silencers, is called the stripe 2 regulatory element, and it's shown here down below. And see, it has a bunch. So this is the stripe 2 enhancer module, regulatory sequences, and all of these genes here, gap and gap genes, they can induce or repress the transcription of the gene. So for example, this module contains elements to which giant and crouple combine, and in doing so, they repress transcription of even skip. And bicoid and hunchback also have binding sites, and they tend to activate transcription. And this element will only lead to transcription in places in the embryo that has low giant and crouple and high bicoid, or high enough bicoid and hunchback. And this is the position of the second stripe in the embryo, and you see we have lots of hunchback, a little bit of bicoid, and virtually no giant and crouple, and therefore the gene is on. So I'm going to stop here. It's really funny, right? So I'm going to tell you that joke again on Thursday, and we'll talk about homeotic genes, and we'll talk about worms, and we'll talk about hedgehog.
I don't think it was.